So if I may, I'm going to introduce David, or ask David Perlin to come back and introduce our next speaker.
So thanks very much, Neil. So in our for our very last talk, we're going to address the issue of solutions for the problem, especially developing new drugs. And when we think about the solutions for drug resistance, there is a universal call for new drugs with novel mechanisms of action that overcome existing resistance. This is the same call that we have for all antimicrobial agents. The push-pull commercial incentives to invest in new drug development are really quite complex. And really in the past 3 decades or so, no one has done more to advance the development of novel antibiotics and antifungals and to address systemic issues that plague development than our next speaker, John Rex. So John, initially as an ID physician and expert in mycotic diseases at the NIH and then Texas, and leading a variety of preclinical development programs, then as a senior leader at AstraZeneca, leading a worldwide antibiotic development program. Then since leaving AstraZeneca, back to mycology and acting as the chief medical officer for F2G, which is developing larafin, which is one of the new classes of antifungals in the aradomide class. John, beyond the development work he's done, he's been just a phenomenal advocate. And he was really the architect and helped co-found CARB-X. And for those of you who don't know it, CARB-X is this private-public partnership to really infuse funds in for antibiotic development with $1 billion or more funds, which has just been an absolute game changer in bringing companies to the table, and probably 9 out of 10 drugs now being developed are being developed by small companies. He's was instrumental in the new drugs for bad bugs of the European Innovative Medicines Initiative. He's served on numerous panels, including most recently the US Advisory Council on Combating Antibiotic-Resistant Bacteria. If you don't follow his blog, you should. And you should read his newsletter. It's really quite both entertaining and educational and important. So without further ado, I'd like to introduce then John Rex, who's going to focus on coming of age and antifungal research and drug development. John?
Thanks, David, for the introduction. And it's fun to be home. Let me tell you, this is home. I'm gonna make that go away. And in theory, you should be seeing a slide saying antifungal drug R&D comes of age. Make a noise, David, if that slide is not showing.
We see them, John. And if you put on, are you in presentation mode?
Well, I'm going to leave it where it is because that way I know what slide's coming. I like having the cue cards on the left, and I think it's readable enough. So there are my coordinates, and happy to absolutely to share these slides. Anybody wants a copy, just drop me a note. So let me just say, it's fun to get to be here. I've been a student in mycology for nearly 40 years. As David says, I've done all kinds of different things along the way. And that's actually what makes me qualified to talk to you about developing drugs, because I have tried, I have been burned, I have I have had the up, I have had the down, I have hit the wall. I've done antifungals, I've done antibacterials, I've done everything from preclinical to marketed compounds. And what I'm going to do today is give you a tour of some of the ideas that I find most relevant to developing new novel therapeutics. Focus is sort of mid-discovery to registration. I'm not going to talk about targets other than incidentally. My choice of topics is completely biased by personal experience and lessons learned. And I'm going to draw some ideas from the bacterial world because there's a lot of similarity there and things to learn. And for avoidance of doubt, David's already noted that I do work for a company that has a compound in phase 2, but I'm not going to talk about it. I'll mention it tangentially at one point because there's an idea that you can borrow and read about our compound. There it is. That's all I'm going to say about it. So the big theme is, what does it take to invent and deliver a new drug? And a new drug is different from a new chemical, okay? And so let's start with the idea of unmet need. Everything you work on needs to have a clear value. It's true of everything from new cars to new iPods to antimicrobials. But antimicrobials, making a demonstration of why something is interesting is more subtle than you might imagine. How do you prove its advantages in the clinic? And we're going to come to that. And it's important that you know now what hurdles to leap, because that may set you up to make better choices. And the best advantages you can put in your compound are things that are durable and visible and permanent, like novelty. Nobody can ever take that away from you. Oral can't take that away from you. Big things that have a lower frequency of resistance, those are all good things. The things that you can choose within limits, you can choose them. Sometimes you just got to take the drug you get, but you can make some choices. And mycology has lots and lots of unmet need. We have relatively few compounds. I really enjoyed Judith's talk today, that nearly empty slide of mechanism of new antifungals, of antifungals at all. Things that are oral, we've only got one class. The resistance is everywhere. There are places where current agents struggle entirely. You can't always cure cocci. I could go on and on, but this lovely graph that Matthew Fisher's group put out, and I believe I saw his name on the call. Thank you, Matthew, for this slide. Decades going down, darker is worse. Resistance rolls on and on and on, and will always be with us. And so we're always going to need new therapies and new approaches. So the thing it takes to invent and deliver a new drug is it takes a village, many, many disciplines, biology, med chem, long list. And being selective, I'm just going to look at a couple of them in some detail. Biology and the role of academia. One of my colleagues, Don Drakeman, wrote a lovely paper in 2014 where he looked at the sources of interesting new molecules, and he defined interesting as FDA gave it priority review. And what you saw there was it came out of your laboratories. Small companies really power the pharmaceutical industry. But the key point I want to make is, once you think you have something interesting, get professional help. Don't try this at home, kids. Get a pro at your elbow, because there's a whole raft of stuff out there that you just have never spent time studying. Okay, and I'm going to point out a few of the ways that you can run into a wall, but only a few. I'm constantly inventing new ways to hit a wall. So toxicology. You need to study supratherapeutic exposures of the new agent and all of its metabolites into animal species. And serious fungal infections often require lots and lots of therapy. So you're going to have to do very long toxicology studies. This takes a lot of money and a lot of products. You want to dose mice for 6 months, you got to have enough drug to dose them for 6 months in a formulation that gets into them reliably. And metabolites can be tricky. You can try to predict the human metabolites, but you don't know until phase 1. So you've really always got to be planning for this one. But the key other lesson about toxicology is don't panic. Everything has toxicity, and your job is to triangulate the exposure you think that's required, the nature of the toxicity, and the unmet need you're addressing. This is your first look at benefit risk. And benefit risk needs to be B really, really greater than R for vaginal candidiasis, but B versus R can be almost the same for, let's say, curative therapy of CNS cocci. And the core advice here is have a PK/PD expert and a medic on your tox team way back at day 1. And there's some talks I'd like you to listen to from a Carbex boot camp. Manufacturing and controls. Oh, this is fabulous. How does a molecule become a tablet or an injectable on the shelf? I'll tell you, I knew nothing about this before I joined industry, and it is a whole science unto itself. You have to plan for the early materials for preclinical studies, GMP materials for human studies, and then sufficient quantity at scale and on stability for phase 3 registration. Get help. That's the message. You can look at these things, but don't try this by yourself. Get help. PK/PD is another area where antifungals have begun to evolve. We've long used PKD, with 20 years we've been using PK/PD in a serious way for antibacterials. We're starting to use it seriously for antifungals, and we struggle because we lacked some sort of good endpoints in animal models. But that's substantially improved at this point. We have been able to develop PD targets for Candida. We've been able to develop good endpoints for the molds, for Aspergillus in particular. So at this point, for a novel product, actually the hard part is defining the PD target that has meaning. And here your PD targets are just a number, and you can sometimes push that number to infinity, and of course that actually leads you into all the toxicities. With a known class, you can lean on precedent. You know, if you're developing a new carbapenem, you know kind of what a carbapenem ought to do. And if a new one doesn't roughly match that, you kind of have a big think. But with a novel class, you know, what do you do? In animal models, you can push drug exposures to levels producing 100% effects your endpoint of choice, but those exposures may not be needed in man, and they may not even be attainable in man. So this is a topic that I could spend an hour on, and that William Hope could spend several hours on. And I'll just say that I like his quote. It's enough when I would be prepared to be the first person to administer a new compound to a patient. The best idea I've got for you is to think about using benchmark compounds in your animal studies to decide when you think you've gotten to enough. And there's a paper that I got to write with George Trusano and William that you can read about that topic. All right, regulatory paradigms. I want to talk about the structure of approval programs, because I think it's really helpful to understand this when you're thinking about how to develop your compound and what it will take to get it approved. There are some compounds where I can envision knowing how to use it if it were on the market, but the question is, how do I get it on the market? How do I provide enough data to show people who needs the drug, how they will benefit from it, and how do you select the people who will benefit from it? So let's look at an example. And here, the best recent example in medical mycology is isavuconazole, a novel mechanism, or a novel azole, not a novel mechanism azole, that has a 2-element registration program. They did one randomized control trial of a non-inferiority design, ISO versus VORI in azole susceptible, IA. And they did an open label study that kind of took a lot of different kind of fungal infections, but the focus was on mucormycosis. And they provided their controls outside of that study. And the questions I want to ask are, why this pair of studies? Why study azole susceptible infections? Why not just study difficult infections? And I hope that about 10 minutes from now, you're going to know the answers to these questions. Maybe less than 10 minutes, because we're going to go take a tour of antibacterial development. And here's a sidebar. We have to realize that all clinical studies reduced to, actually all studies reduced to one of two types. It's either a superiority study where you're showing that X is better than Y, or a more difficult study called non-inferiority study, where the goal is to show that X is about the same as Y. Now, superiority is always preferred when you can do it, because it's intrinsically validating. If you show that X is better than Y in your study, you've shown X is better than Y, you've shown that you know how to measure the benefit of X over Y, you've shown that you know how to select the patients in whom X is better than Y, and the results are unambiguous. X was better than Y. I love it. When we're developing new antimicrobials, we are almost always forced to do a study where we need to show that X is about the same as Y rather than showing superiority. And this is a very painful idea. The paradox of antibiotics is that while we want new drugs for bad bugs, and while the superiority of new is easily shown in the lab on the basis of MIC testing or in animal models, asking for clinical data leads to a problem, because trials have to be designed usually to avoid superiority. Instead, we have to use this non-inferiority design where we show activity similar to that of another active agent. Here's our thought experiment. You personally have a limb-threatening infection. Your leg is red hot with MRSA. It is, and I come to you and say, I've got a new study and I want to study methicillin versus my new drug. You say, but wait, methicillin won't work. I say, yeah, that's all right, but you got to show superiority. You say, no, this is my leg. I want a cure. Exactly. No one wants to be in the methicillin arm in that study. They want to be in the vancomycin arm or the linezolid arm versus your new drug. That they could deal with. We don't enroll people if the either new or the comparator looks like it is unlikely to work. And I'm going to tell you, we can spend lots of time debating about this. You know, in other settings, you can do placebo-controlled studies. Placebo, like a migraine. I can do a placebo-controlled study once in somebody with a migraine. In cancer, placebo is not possible, but there's always room to improve on 5 or 10-year survival. But in infections, we do something interesting. We produce a cure. And it's hard to improve on cured. Once you're cured, there's no more cured. You're cured, you're done. But this idea of non-inferiority, God, is so tough. I want a better drug. The thing you have to realize is that insisting on clinical superiority before approving new drugs means progress only when and if the pipeline again becomes inadequate. There's a whole video on this on the website, non-inferiority versus superiority. Go check it out on the YouTube channel. Listen to this if this idea is not familiar to you. The key is that NI studies let us develop drugs before we need them. So what this means for antibacterials is that we have a sort of a template. What do? Got a new antibiotic. Well, you do one non-inferiority randomized controlled study versus a good comparator. And you do it in what we call the UDR, the usual drug resistance setting, which means it's not XDR, not MDR. It's a place where there is a good comparator. It's the usual stuff. Both agents are predicted to be active, and you do it in one of the major infections like complicated UTI. And then on the side, you do a all-comer study for highly resistant pathogens, and you randomize, if you can, to best alternative therapy if it exists, open label if the N is too small for that. And an example of that is the registration program for plazomicin. I could have picked any of several other drugs. This is what you do. You do one study, a highly resistant study, and one complicated UTI or similar study. Let me make a comment about narrow spectrum agents. I do get asked about this. This is the idea that's sometimes called tier C pathways, where you're looking at a super rare pathogen where you can't even do a randomized study because there's so few of them. That's an interesting idea. It might be possible, but do not think of this as simpler, faster, or cheaper. It is not just a regulatory hurdle. When you can't produce the typical quality of data, lots and lots of people ask questions about it. And it's not just the regulatory agencies. Everybody's going to ask this question. If you have a narrow spectrum agent that can't be developed outside of a teeny tiny population, read these papers, give me a call, because it's an area where it's tricky water, and I'd like to see you succeed, but it's easy to screw it up. Another area that I get asked about a lot are agents that augment, perennial topic. I can say I love the idea of like a virulence inhibitor, immune activation. I think that science would be so cool. The problem with developing them, though, is that they're add-ons. They are not therapies by themselves. You're not going to be willing to treat a new something with just an antitoxin, with just a virulence inhibitor. You're going to want a drug that acts directly on the microbe as well and acts quickly, which means the base therapy needs to work. And maybe you can protect a base therapy from emergence of resistance, but that still doesn't solve existing resistance problems. I'll just comment that lack of an MIC, a lot of these products don't have an MIC, a little harder to apply PK/PD, but that's probably solvable. The thing that's not solvable is the superiority problem. You're going to need, at the end of the day, to show that new plus old is better than old. You may need a new novel endpoint to show value, and you hit a math problem. And the math problem is that when you start to think about showing that I'm going to improve on standard of care, improve on SOC, standards of care are actually generally pretty good. When the standard of care is like 85%, 90% perspective, you've only got this tiny little increment on top in which new can produce an increment. And that's really hard to measure clinically. And if you do a study and say, well, the response rate went from 90% to 92%, say, well, do you really believe that? Does that really matter? That's the hard thing about all of these add-on agents, is that you've got to find a place where there's actually a big gap. Endocarditis might be an example. Some fungal infections might be an example. You might need different endpoints. And again, I'll say this is not a regulatory problem per se. The agencies are just the first to point out the issue. Everybody wants to know, why should I use this and why should I pay for it? What's the benefit I'm going to get for my money? So now we're back to this. Why did the developer of isavuconazole do this pair of studies? Well, in the UDR setting, it was possible, took some years, but it was possible to do a study in azole-susceptible aspergillosis versus voriconazole. They got it done. They got a clean result. ISO works just as well as Vori does in azole-susceptible aspergillosis. And the other benefits of ISO are then available to you if you want to use it. Mucormycosis, open label study, some control groups. They showed, looks like this is actually at least on par with an amphotericin. It's certainly a lot less toxic. So that's interesting as well. So that's the idea of what you're able to do. And if you just wanted to study resistant pathogens, well, you'd still be doing this program, okay? You would never have finished. And studying resistant pathogens takes a long, long time to get done. So future agents are going to need to apply these tools. There may be a little more scope for showing superiority with antifungals in some settings, but generally it's a tough road. But requiring clinical data on superiority, that's clinical data, for every future drug creates a catch-22 that must be avoided. So now let's talk about avoiding that catch-22. So really shift gears here. We're going to talk about payer paradigms. Why should anybody pay for your antibiotic, and when should they pay for it? And it's important to recognize that the current approach to antimicrobials in general, this is true probably as well as, maybe a little less so of antifungals because we've got so few of them, but it's still going to be true of antifungals, is that everyone is delighted to have a new antibiotic. You bring it on the market, you get lots of pats on the back. Well done, young man. Glad you got that on the market. That's an important new drug, and it's so important we're not going to use it, which makes perfectly good sense from a stewardship perspective. It's exactly what we should be doing as a community. But in terms of revenue, to pay for the development costs and to keep the lights on and to manufacture the drug and to do the pediatric programs that everybody would like done, it's a wipeout because there's no money coming in the door to do those things once it gets on the market. As a consequence, we have had several companies bring new antibacterials to market. Hooray, got approved. Boo, bankrupt. And the problem is that the current pay-per-use model, that is pay only when we have a patient who's got an infection, reimburses for only a piece of the value of a new drug. So, pop quiz. We're going to talk about a related idea. And you'll have noticed the little icon in the upper right-hand corner. Why do I have a fire extinguisher on all of my slides? So here's your pop quiz number one. Have you used a fire extinguisher today? Now, what you're thinking is saying, nah, I haven't used a fire extinguisher today. I'm going to ask you again. Are you using a fire extinguisher right now? You might say, why does he keep asking this question? Well, it's because, in fact, you are. Are you in a building? Are you in an apartment building? You're anywhere near any kind of real estate at all? I guarantee you there are fire suppression systems around you, including an entire fire station. And the only part of that system that you're not using is the part that gets you wet. All the rest of it is what makes it safe to be in that building, and you know that, and that's why you're willing to be in that building. And the theme here is that the fire department and the fire extinguishers have a value simply by being present. The fact that I have a fire extinguisher under my stove means that if I set my lunch on fire, I can put out the fire and the building doesn't burn down. I want that fire extinguisher. And the fire extinguishers, by being present, can make you safe in your community, in your hospital. And one of the ways to summarize it is this acronym that we call STEADY. And I'd encourage you to read about the idea of the STEADY acronym, or just videos to look at. But STEDI stand for the non-use values of antibiotics. Let me just pick one, transmission. If you have an infection and I treat you, then the person next to you can't get that infection because you're no longer transmitting it. That person has gotten real value out of the fact that you were treated. So that's the transmission value of an antibiotic. Might mean that I get to keep my ICU open because I can treat the patient before the infection spreads. That's the transmission value of an antibiotic. And how do we reward that? Because we're really used to paying for new drugs only when we use them in a human being. Well, this is where we say antibiotics are the fire extinguishers of medicine. You have to buy them the way you do fire extinguishers. And that's led to a lot of thinking about new incentive models. If this is of interest to you, I encourage you to read some of these reports. You can go to also to the website, just John Rex AMR incentives will take you to a number of recent papers. The concept here is that after lots and lots of thinking about this, we actually spent 3 years in a project in Europe enumerating all the possible ways to fix this economic problem. Came up with a list of just under 40 possibilities. Many of them were, yeah, it's true, but that would never work in the real world. There were 4, however, that came through at the end that said, these 4 have the least warts and the most punch. Those 4 are, 2 of them we call push, which is just money to encourage people to come do stuff. Grants, coordination of grants. We're doing that through programs called like Carbex and Novos Repair and NIAID. Pull is the thing I want to talk about, is the label for how you pay for antibiotics as if they were fire extinguishers. And there are 2 kinds of pull ideas that require just a tiny bit of explanation. So the first one, we'll call this big pull, market entry rewards. The essence of the solution is that if you invent a really interesting new antibiotic, we, the community, via our governments, will agree to pay you a certain amount of money per year for a period of time just to make that drug and have it in the pharmacy. You can still sell the drug a little bit on the side, we suppose, in some circumstances. In other circumstances, you don't sell it at all. But the concept is, really, you're not worried about the volume of sales because you receive a certain amount of money, predictable, based on the quality of what you invent. And that's actually 98% of your commercial model. The market entry reward does the heavy lifting. Please go watch the video if you want to learn more about that. The other model is the fix for what happens after 10 or 15 years. The drug is now off patent. We've paid back the people who did all the work, and yet we'd like to have the drug for the next 30 years. I'm only going to use it a few times a year. Penicillin G for neurosyphilis. I'm not going to use it very often, but when I want it, that's what I want. Who's going to make that drug? And there was a nice review article by Celine Pulcini and colleagues that talked about the fact that antibiotics disappear. Some things you can't get. Well, why is that? Pop quiz number 2. What does it cost per year to maintain the plant that makes a sterile injectable so that you can have at least 1 vial a year? Probably make up several 100,000 vials a year, but realistically, make some, run the plant, pay the staff, do all the paperwork, blah, 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 blah. What's that cost? Well, my rule of thumb is that costs about $25 million a year just to keep the lights on in that plant. And if you want to have penicillin G available to you, somebody's got to run the plant. So long-term continuity is about just having the idea of, we find some contract manufacturers and say, would you please make this stuff? Just make it. Whether we use it or not is our problem. Just make it. Sweden is now testing a model like this that pays for access. So putting it together, there are a lot of puzzles still to be solved. We don't yet have a viable market entry reward model, and we're working on questions like, what's the value? Do all antibiotics have the same value? The answer is no. Who will pay? How will the payment be delivered? Well, the UK is experimenting with it. The US is now experimenting with it. The EU has said they're going to experiment with it. And there's some things you can predict about how this is going to come together, because I think this is the future. So what does this mean about your choice of development projects? Novelty, novelty, novelty. Fire extinguishers come in different categories. At the end of the day, you only need one of each. Incremental extensions, some, but only get you so far. What you really want is easily articulated scientific value that no one can take away from you. And if you look at the NHS, the UK NHS pilot model, they've laid out a point scoring scheme. You get a certain number of points for novel mechanisms, a certain number of points for covering certain bacteria, a certain number of points for certain resistance mechanisms, novelty, and demonstrated scientific value. And so the implications for medical mycology, the focus in all the prior work, much of the prior work, has been about antibacterials. That's the thing that's burning a hole in everybody's pocket. But all of us on this call recognize that antifungals are, I like Tom Chiller, I like your line. Antifungals are antimicrobials too. Yes, don't leave us out. The only fungus-focused elements are in the US so far, where the GAIN Act, QIDP, and LPAD have all recognized the unmet need for fungi. Hope to keep fungi in the Pasteur Act in the United States, which is what's the pull incentive in the United States. We just need to keep at it. You know, all of you now are a bit more aware to harken back to Mike Rinaldi. I hope you're a little bit fungitized about how you're going to talk about fungi going forward. And being involved with lobbying groups, which is something I never intended to be doing in my life, but that's actually part of what I do now, is important. So in summary, here's my advice if you're interested in developing a new antifungal. Start early, get professional help. Your academic idea is the heart of it. What you need are people who have built the bridges, who have driven down that road, who know how to do all that other stuff that you've never studied. Study the antibacterial literature. Attend or at least listen to as many FDA advisory committees as possible. The isavuconazole AdCom is a good place to start, but we don't have that many for antifungals. So, do the others. I will say FDA had 3 antifungal workshops last year. They were great. They're all available. They'll replay. Go listen to those things. These are master classes and they're free. Focus on strong approaches to unmet need. Do not waste your time on minor improvements. And the newsletter that I send out is a good way to hear about current events and kind of stuff that's going on. Into the deck, there's a whole bunch of read, listen, and learn stuff. And thank you for listening.
Thanks very much,
Stop
John.
sharing. There we go. Okay.
If you stop sharing, then I think we can get back. Perfect.
Bingo. I found the right button. Took me a second.
So we do have some questions. I'd actually like to take the first question because I want to get at this last point that you were making in terms of the push-pull incentives. So if in the antibacterial space, 9 out of every 10 drugs now are being made by small companies, how are we going to do that in the mycology field where companies, there is not as much investment in small companies, capital markets aren't quite as receptive to be able to create that, because big pharma has basically said, we really don't want to develop antifungals.
Well, I think you need to get your head out of big pharma. It's because I think one of the things that a good pull incentive would do, let's think about this, the UK NHS mechanism. The UK has said that a good antibiotic ought to be worth about somewhere in the range of $2 to $4 billion over 10 years. And the Pasteur Act of the United States is saying the US thinks that a good new antimicrobial is worth between 750 million and 3 billion over 10 years. So I'm just going to pick a number. Let's just say that let's suppose that in some future universe, 5 years from now, it is possible to predict that if I invent a new drug that has these features and I get it to approval with those things in the label in some fashion, I will receive $2 billion over 10 years as the delinked market entry reward for my new drug. That's my market model. That's actually, if I've been clever about my investments up until now, if I've kept my costs down, I can actually return my cost to the investors. It actually makes it as logical to invest in me and my new idea, because I can say, look, if I get there, and there's always technical risk. Pharma understands technical risk. What we hate is the reimbursement side. I want to know, can I generate the income? I'm saying, look, here it is. Here's your number. There's your number right there. That actually is and should be very powerful across the community. And we need to get antifungals on the list with antibacterials. They're sort of on in the United States. They're kind of wiggling their way in. So that's what I'm hoping we'll see happen, is that this will actually transform the landscape for investing in a new company.
So we have, thank you. So we have a few questions. So Tahana wanted to know that as you're developing a new molecule, is it important to demonstrate that it has a low propensity to develop resistance, and how would you go about doing that?
Yes, and whatever you think is best. I mean, I'm not going to write down a particular answer to that. Somebody asked me earlier about the previous conversation about when do you put that into your programs, this resistance thing. You got to take yourself back to, I said I wouldn't talk about early discovery, but I will. At the very beginning of time, when a new project, the chemist has gone back and says, look, here's my sample. How much is there? Well, there's 10 micrograms. Oh, okay. What am I going to do with that? Well, I can test that in 6 things, sort of, maybe if I dilute it carefully. And in the make test loop, the chemists are constantly evolving, and every milligram is expensive that you make. And so the best thing that you can do early on, I think, is pick out targets where if you're target based, maybe just know that the target itself is intrinsically reasonably well preserved, because you're not going to find out until much later what the true propensity to develop resistance is. But you can work that in down the stream in your make test cycle. But early on, all you can really do is just kind of triangulate on the best available information. David, what would you do?
Well, we'll have to talk offline about what I would do. Coming back to push-pull incentives, so Mike Bromley wanted to know about rare fungal diseases, like developing nicomycins. I mean, is that something that could be done using these models?
Well, the push-pull model, the push part, of course, is grant money. Anything can drive that. What we need for the pull incentives is to have them linked to and reward antifungals. And I think you really got to separate out a little bit the problem of the technical hurdle of developing drugs for certain circumstances. The less common the disease, the harder it is to do the work, and that's really just sort of the fundamentals. If you're gonna, if we're all very interested in antifungals here, but the truth is they're not nearly as common as bacterial infections. Think about the isavuconazole IA study. It ran for 7 years to enroll those. It actually enrolled 500 patients over 7 years. Now, there was a little break in the middle. They had a supply chain issue, but it's still a long study, and you've really got to want to do it. I think that is unlike doing complicated, in bacteria, complicated UTI, God, you can do a 500 patient complicated UTI study in 18 months. It's just night and day in terms of developability. That's a message we have to give to people, because Chiller's got these fungi on his watch list, right? And he's saying we need new drugs for these things. Well, if it's impossible to develop them because we set our standards so high, that doesn't do anybody any good. But we can't set it, but our standards have to be high enough that you actually believe the drug works. So there is this balance, and I will say the agencies are generally quite willing to talk to you about that balance if you can articulate it. And there's this whole pathway in the United States in particular called the limited population antibacterial and antifungal drug pathway. Now, it's usually, you'll usually see it as LPAD. People think limited population antibacterial drug. It's actually LPAAD, so that would be LPAD, but we abbreviated LPAD. So the limited population antibacterial and antifungal drug pathway says that when the thing you're pursuing is super, super, super rare, and you can identify the limited population of people who really have it, that's the key. You can absolutely say, this person has it, that person doesn't have it. We're not going to be passing this out at CVS or Walgreens. Only in this limited population are we going to use it. The FDA has a lot more flexibility. So and that's a concept for rare fungi.
So another question is, can you use these development principles for vaccines? What are your thoughts about fungal vaccines or even biologics? So
I am modality agnostic, so I don't care whether a therapeutic is a biologic or not. It's still just got to do something. So, but vaccines, God, you wish the vaccines were easier to work with, but for good reasons, I suppose they're not. So there's no reason whatsoever why you can't develop on paper a vaccine for fungal infections. They're just saying no one's going to stop you. If you can identify something that prevents invasive candidiasis, that prevents invasive aspergillosis, rock on, okay? Your problem's going to be proving to me that the thing that you've developed actually does that. Because no one's just going to say, well, you created an antibody. Well, nobody cares about that. What they want to know is, did it prevent infections? And let me just give you a salutary story. How about Staph aureus? Really common bacteria, right? You might think a vaccine for Staph aureus would be a straightforward thing to do. We know a lot about it, need it. And yet developing a vaccine for Staph aureus has been wildly difficult. In part, there's a biologic hurdle. But the issue is that you want a Staph aureus vaccine to prevent Staph aureus infections. How are you going to do that? The clinical trials to show that you're going to prevent infection require you to take people who should get an infection and reduce the rate. Well, we do lots of other things to keep the rate down. You say, well, maybe I'll work on hip replacements. I'm going to prevent Staph aureus infections after hip replacements. I can tell you that what it would take is a study of about 50,000 people to show that you reduce the rate of hip infections with your vaccine from 0.25% to 0.15%, and nobody's really going to care. It's way too big. The number needed to treat is gargantuan. So your problem for Aspergillus in Canada is that you've actually got to show me that it does something. It's got to prevent infections. Okay, here's my vaccine that prevents IA in people who got a stem cell transplant. You do that, you roll enough people and show me the number, everybody's going to cheer for you. It's just really hard to deliver.
So, in the past 9 months or so, we've had all these emergency use authorizations for COVID-19, for things like remdesivir, for biologics, antibodies, convalescent plasma, and so forth. And you were talking about PK/PD. It was sort of non-existent for a lot of these agents and probably reflects on some of the efficacy. Nevertheless, we've used this mechanism, the emergency use authorization. Alejandro Pasqualato is asking, is it really necessary to do a phase 3 trial? I mean, can we just say, the phase 2 looks really good, do we need to do a phase 3? Do we need to spend all that money and time if we really need to get something out? And should we even consider an emergency use authorization for certain agents?
You know, that question does come up from time to time, and the answer is, you know, if the circumstances are right, clearly the FDA will engage on that. There's no doubt about it. But you're not going to do that for something that's not as big a public health emergency as COVID. I mean, we expect our medicinals to have certain properties. You got to know that the manufacturing is stable, that you know, every time you get a pill or a tablet or an injectable, it is what it says it is. You've got to know what the safety profile is, and your willingness to tolerate absence of data is inversely proportional to the size of the blazing fire. With COVID, you know, it was all hands to the pump, and we also had lots of sadly lots of patient material to work with as possible to do some work promptly. But you just, you know, realistically, the answer is we're not going to just approve things randomly because you've got a little snippet of data in phase 2. There are too many examples of, you know, I have data on 3 people that didn't pan out in larger studies that for anybody to be willing to do that.
Yeah. So Tom, in the US, we rely on the CDC to tell us when we have an emergency, what's critical, what's not. So, you know, so how are things organized at CDC in terms of advocacy, and, you know, how do you decide what's an emergency and what's not?
Good questions. It's been good listening to John talk. I enjoy his perspective and have for many decades. So thanks, John. Good to hear you. You know, we have a small group of mycologists at CDC. I think there's around 35, 40 of us, give or take, students and fellows coming in and out. And so our job is to advocate for public health mycology. And so in our branch and within our organization, that's our job. And so what we try to do is, you know, really reach to all aspects of the agency, both up and down, and you know, insert ourselves into initiatives like AMR, like advanced molecular detection, like advancing surveillance of hospitalized data and patients. And in all those different areas, you know, our job is to advocate that fungal infections and diseases are included. When it comes down to defining urgent threats and problems, I think we all probably carry our own flags to a certain extent, and we all get in the room and we try to advocate for why our organism should be on certain lists or not on certain lists. And, you know, I think what it breaks down to is a few things. You know, I always like to say, you know, how deadly is this thing? You know, are people dying? Because to me, my ultimate goal about working here in public health is to save lives. So I feel like if I'm not coming to work and thinking about saving lives every day, then I should probably be looking for something else to do. And so I tend to look at mortality, look at death, you know, look at those kinds of infections. And so, you know, I'm often pitted against my colleagues, for example, in salmonella, trying to argue what's more important, salmonella or aspergillus. And I will argue, well, to me, there's sort of a no-brainer there. I mean, salmonella kills 300 people a year and aspergillus kills thousands. And so I think we need to, you know, definitely aspergillus needs to be on that list if salmonella is up there. And so those are some of the arguments I make. And then of course we look at cost. And the challenge with fungal diseases, as we've talked about, is advocating for their importance based on burden and cost is challenging because the estimates are guesstimates at best. And I know we're way underrepresenting those numbers, you know, in the US and other countries as we try to estimate. So short, long-winded answer to your question, David, is those are some of the things that we look at in how we're organized.
And let me just add that if you think about this is a compilation of all the priority pathogen lists, and CDC's the 3rd and 4th columns over, and the fungi, well, we'll find them. There's aspergillus down there, and there's Candida right there. But boy, there are a lot of bacteria that are a problem, and that's, you know, it's just going to say, you know, that's another share of voice is an issue, if you will, in this space, is that, you know, there is a balance. There's only so many dollars. There's only human beings that can do this work, and so, I want to see more antifungals work, but I also know that Acinetobacter kills people too.
So, John, coming back to the question of combination therapy, even developing new drugs and combining them with registered drugs. So, what is your view of that? Should this be a goal to be able to limit development of resistance?
Well, separate the two ideas. First is, would my initial approval be for my drug as used in combination? That's actually a much harder pathway than an initial use as a standalone agent. You can do it. You certainly do it in HIV. Did it recently with a new TB compound. Your TB compounds can only be used in combination. But there are issues around doing it. And the biggest one is with the new chemical entity, you've really got to get a clean safety database on that. That's one of the biggest challenges of the new chemical entity, enough safety data. And to do that in combination, it muddies the waters. That's one of the hurdles you get. But once it's available, the idea of all of us in infectious diseases we've long used one agent, we often use one agent at a time. Well, is that really the right thing to do? Should we be thinking about making combinations? In TB, we've been hit over the head for years. Never use less than 2 active new drugs. So start with 4. So should we be doing that in other spaces? Part of me says that would make sense. On the other hand, it would cost more, and you'd get all the toxicities of all the drugs at the same time. There you go.
So I know we're coming to the end. So just one last comment from both John and Tom. So can you tell us what the next 5 years will look like?
No.
Tom?
Yeah, I think the next 5 years we will be doing more combination therapy up front. How's that? I agree with John that it's, as a clinician, I just don't understand why we're not doing more of it. I feel like I do it in every other infection I treat except for fungi. But and then secondly, I use this thing all the time. You guys are the fungal community, so you know this. But when you have John Taylor and others estimating there are 5 million fungal species in the world, and when only a few hundred of them cause human disease, maybe a few hundred more cause animal disease, maybe a few thousand cause plant diseases, there's still a lot of room for growth out there and emergence. And I look at that and I'm concerned about that every day. Candida auris has really caught us by surprise here. This now potentially pan-resistant organism that will spread in healthcare settings that we weren't thinking of that. The other one that's on my radar is Sporotrichosis brasiliensis, which spreads in both forms. It's a dimorphic fungi, but it spreads in both yeast, and mold. And that's never happened in a dimorphic before. So that's concerning. These fungi adapt, they're smart, they've been around a lot longer than us. And so I think we need to do as much as we can as a community to attach ourselves to other communities and to make sure that we're part of the discussion of AMR, of infections, of emerging infections, and not excluded from it. And so, yeah, I'm looking at the next 5 years as to making more leeway and headway in that, but also being on the lookout for these surprises.
Yeah, so the surprises are already there. Aspergillus. Somebody talked about all the cryptic things that we didn't used to know were there, and now all of a sudden there's all this stuff in there that nobody recognized.
So on that note, I think I'll turn it back over. I want to thank Tom and John, and turn it back over to Neil.